skills in this practical that you expect to see again for your actual practical. Yeah? I'm already telling you up front, you expect to see the same skills. Okay? So microscopy, table drawing, graph drawing. Right? Okay, come. The first page 3, let's talk about table drawing. Alright, table drawing, 5 marks. What does each mark go to? First one, T for title. So first skill, table drawing. Okay, first and foremost, you need to have a title. Your title needs to come with units. It should also come underlined. Okay. If you didn't, then you would have gotten the mark for table. Okay, sorry, for title. Next one, headings. H for headings. Okay, for headings, you also need your units. So for example, in this case, if you wrote time taken, it should be in seconds. Then you don't have to repeatedly write seconds anymore. Okay? So for example, time taken equals seconds. Some of you wrote SEC. Okay, this is not the unit for time. Yeah? So that's heading. Next, C for column. How do you spell column? Should I spell column? Column, Okay, for column, what are we looking out for? Number one, then you eventually draw the table. Okay, so for example, there's no table here. Okay, so it's just values written in straight rows. Okay, so make sure you have the table. Then it's drawn with ruler. Okay, your table should be in pencil. Table with ruler and pencil. Okay, next, your independent variable should be in the very first column. Don't know what independent variable is? Okay, I just write first up. Independent variable should be in the first column. Okay, what is independent variable? In this particular example, the independent variable is the percentage of Prophase which you were changing. The dependent variable is time. That's what changed with the change in concentration of Prophase. Okay, so independent variable is the thing you change. That should always be in the first column. If it's not in the first column, also you won't get the mark. Okay, V for values. Okay, your values, please follow instructions. If you read the very top, they say, construct a table below to record your results to the nearest second. Okay, it says to the nearest second. So your values need to be in the correct DP. That depends on the question, okay, whatever the question says. It depends on question or the measuring instrument. Okay, finally O for observations. For your observations, as long as we observe some sort of a trend, we will give you some mark. We will give you the mark. So you need to see some sort of a trend. Don't leave any value, any row blank. If you find that you have no time to complete your experiment, what should you do? I didn't say anything. Okay. But make sure it follows some sort of trend. Okay? Yeah? But do your best to finish the practical. Okay, plan your time well. Because you never know. Imagine the trend is that it goes up, then the last one or overseas give you something that dips down. So you never know. But in your desperation, just don't leave it blank. Okay? Okay, that's for table or drawing, 5 marks. Next, the one that is very heavy in marks is biological drawing and graph drawing. In fact, biological drawing weighs more. So let's get down to that. Biological drawing. Okay. Wow, I don't know anything. Okay, first T for title. Your title should be underlined. When in doubt, just underline. Okay, it should be underlined. No underline also, we won't give you the title mark. Make sure it's underlined. Also include the magnification that you are viewing at. For example, do you view at times 10 or times 40, etc. Okay? Title. T, then C. Okay, C for clean lines. Okay, no hairy lines. Unless it's actually hairy. Okay, no hairy lines. Okay, no gaps in the lines. That's what clean lines should be. Your drawing should also be in pencil. It should not be in pen. L for labels. Your label, the line should be in ruler. A for accuracy. Okay, A is the most dubious one. I honestly don't know what you saw. So whatever you drew, right, I'm going to take it as accurate. Yeah? Okay, but this is something that I usually won't let my cell wall. Your cell wall, if you have a plant cell, if you are given a plant cell, listen up. If you are given a plant cell, make sure that. Okay, so under usually accuracy mark, one thing I am looking out for is that it takes around 3/4 of the space that we give you. Okay, takes up 3/4 of space. Other than that, I'm looking out for whether the drawing is accurate. The only thing I look out for is the plant cell. Plant cells, the cell wall is not as thick as you think it to be. It's usually very thin. Okay? If you draw a cell wall that is this thick and fat, I will not let it go. Okay? So cell wall. Okay, this one if you say cell wall, I'm okay. Yeah, it's not usually so thick. Next, P. P for parallel lines. Okay, what are we looking for in parallel lines? Please take note, I think a lot of students didn't get the full credit for P and M. Okay, so for parallel lines, this is what you're supposed to do. I'm going to do to the left side. Let's say you have a drawing. You are supposed to do a dotted line. Two dotted lines that are parallel to each other. Then you should have two measurements. The first measurement should be, I'm just going to draw a line up. The first measurement should be, length of actual specimen. Okay, sorry, too small. I think the back can't see this. Do a bit deeper. Okay, so two lines. The first line will be length of actual specimen then you dot dot, then you write the length. In whatever units, maybe mm. Then you have to draw another one. You make sure you have another measurement. So two measurements. Most of you only have one measurement. Two measurements. One is length of actual specimen. This one you need to refer to the microscope. You have to find out using the microscope. Right? Every time you look in the microscope, there's a small ruler inside. Each ruler has a unit. The unit changes depending on which magnification you're looking at. Okay? Then the second measurement will be length of drawing. Okay, then you dot dot, whatever the length of the drawing is. This one maybe is in cm. Okay, so two measurements and two parallel lines, and then you get the full credit for P. The last one, M for magnification. Okay, you need to then calculate magnification. How to calculate magnification? You will take length of drawing divided by length of actual specimen. In other words, magnification is a measure of how much bigger is your drawing compared to the specimen. That's what magnification is. Make sure that the unit is the same. So if it's mm on top, below needs to be mm. If it's cm on top, below needs to be cm. Okay? A bit wasted. I don't see many students doing magnification. One, two, three, four, five, 6 marks here. Yes. For your labels, if you know there is a plant cell, okay, if you know there is a plant cell, it should have a plant cell features. So you should have things like cell wall, cell membrane, chloroplast. If you on the surface you really see it. Okay. Yes.
The magnification, do we just write it aside?
Oh, where do you write it? Is it? You can do your calculations below the drawings or the side of the drawing, doesn't matter. Anywhere in the space provided, you can do your magnification anywhere. Okay, I'm going to erase the left hand side. We're going to go into graph drawing. Everything starts off with title. So it's really a waste if you don't write down title. So go for it. Yeah. Oh, okay. You do it to 1 dp. Okay, do it to 1 dp. Okay, next graph drawing. Are you okay? You need anything? Alright. Finally, graph drawing. Graph drawing also same point. First one, title. Notice everything needs title. Don't know if you need title or not, just draw a title, right title. No loss. Okay, title again needs to have units. This is no different from everything. Okay, units as well as needs to be underlined. Yeah, always the same thing. Next, for scale. The scale needs to use up 3/4 of the space or so. So make sure whatever scale you use, the graph needs to maximize the space. Okay? The scale also cannot be odd scale. No odd scale. I think I went through this before. Let me give an example again. Imagine you have a lot of lines, right? Graph paper come in many grids, right? Let's say you decide that for every 3 small squares, you're going to label it as 1. Then the next 3 square will be 2, and so on and so forth. Then I ask you, what would this unit be? It will be 1 divided by 3. Then it's like, it's a recurring number, right? Yeah, 0.3333. So this unit now will be 0.333 and so on and so forth. That's what an odd scale is. Okay? If you want to know if your graph, whatever scale you've chosen is an odd scale, you will take your smallest unit, go and divide by the number of squares that you've chosen to represent. If you find that after you divide, the number goes recurring, then it's an odd scale. Okay? That's how you check. Next, A for axis. All we are looking for is that you label the axis, label with units. Okay, if you label with units, you're good to go. Okay, next P. P for points. Make sure you plot all points with this symbol, X. You don't use a dot, don't use a plus. Yes? Oh, okay. Axis, if it's slightly shorter, is okay. Yeah. But the title, you try and max it up. Yeah. Be elaborate. Okay. The axis, can you draw your axis lines? Don't use the edge of the graph paper as the axis lines. Okay, so make sure you have axis line. Label with unit plus axis line. Yeah, correct. So if it is the end of the graph paper, just draw one line down, just draw one line across. You don't leave it. And then you also need an arrowhead, right? So whatever axis line you draw needs to come with arrowhead. Okay, so points, all points plotted correctly. All points plotted with X correctly. Must be correct. Okay, teachers will check if it's plotted correctly. You can expect to find graph questions also in your mid-year exams. It may come out also. Okay, it may come out. Okay, very finally, best fit. For best fit line, usually it's a curve or a straight line. The only thing I always look out for is for best fit line, no extrapolation. Then you're quite good to go. I find for graph drawing, personally, I find it the easiest to do because the first 4 points is like giveaway. So your last point, you can actually get 4 out of 5 without drawing a graph if you think about it. Yeah. Okay, so graph drawing is usually my favorite one. No, actually I enjoy biological drawings. I like to draw that. So it's my favorite. Okay, so you see table 5 marks, graph drawing already 5 marks, this 10 marks. Then biological drawing 6 marks. You just do all the skills right, it's 16 marks already. 16 out of, this is 20. You can actually get 16 just by doing the skills. Yeah.
I think you want my axis wrong. I thought the same thing as survival line wrong.
Oh, okay. Oh, I'm a bit wrong because of the you draw the line. Yeah. So there's no axis line here, there's no axis line here, and then also there's no skill. Okay, any questions? Actually, if you zoom in close enough, you usually can see. Or you find another sample. You scroll to another cell and try to find another cell with cell. Usually it's there. You just need to look carefully. Yeah? Oh yeah. So what's the unit for magnification? So it could be 200 times. Yeah. Okay, so but see what you don't see a cell wall? Actually, a cell wall is always there. So this is what it will look like sometimes. You may see a cell like that. And then you see, okay, you just see a cell like that. It looks like it's just one layer, right? That's all you see. Actually, if you think about it, if I want to draw this thing out, I can draw it this way. I actually have an inner layer, and then I have a very thin outer layer. This black part over here, if I wanted to, I can break it out in two lines to represent the thick, this black line over here. So very often when you see this, actually you are looking at a cell wall. You just need, the cell wall is just rather thin. It's not as thick as you imagine it to be. Any more questions for your practical? Yeah, so as you can see, 16 marks are just skills. 4 marks are the tougher questions. Okay? 16 marks of just pure skills. Yeah, okay. What specimen? I mean, Danya didn't need to draw so many. Oh, okay. Yeah. Okay, so for this particular practical, what they wanted you to draw was 3 cells that are side by side. How are you supposed to do that? That's what the question says. The question says, make a large drawing of this group of 3 cells. Yeah? If you see the nucleus, you draw the nucleus. If you see a cell, you draw the cell wall. Yeah, but yours you totally didn't label anything. You didn't even label cell membrane. Yeah? No labeling at all. Okay, so that's your practical. Let's go into your RE. Yeah, apologies as we come closer to exams, we spend more time just discussing about our learning and our work. How's your preparation for media? I understand you only have 1 week to... Do you all have a schedule planned out for yourself? Okay, but I feel personally, first step is always to have a schedule of some sort. Like today, I need to accomplish finishing revising 1 chapter of bio, finish a little bit of math, etc. Okay, when you don't have a schedule, your time just slips through your fingers. It's magical. Okay, so make some sort of a schedule, maybe on your phone, it will help also. Okay, I'll give you the MCQ answers. Only questions 1, 3, 4, 5, and 6 will be done. Everything else is Mitosis. Okay, so questions 1, 3, 4, 5, 6. Okay, the answers are B, D, A, B, D. Right, which question should I go through for MCQ? Question 1. Right, question 1. 4. Anything else? Question 5. Wow, I'm going to get a big bowl soon. Come on. Any more? Okay, don't do it on purpose. So question 2, 3 and 6 are okay? Okay, your class has a different set of questions. Question 1. Which of the following statements regarding DNA replication are true? Okay, according to the answer, statements 1 and 4 are the only true options. So number 1, the resultant daughter DNA molecule contains one parental strand. It is true. It is true. The resultant daughter DNA molecule contains one parental strand. This is semi-conservative replication. So one will contain parent, one half is parent, one half is daughter. RNA polymerase adds in a short RNA strand to serve as an anchor. Okay, what's the issue with this statement? DNA polymerase adds in a short RNA strand to serve as an anchor. It's not DNA polymerase either. What adds the RNA? Yeah, okay, it's primates. So statement number 2, the issue here is primates. Primates adds a short RNA strand to serve as an anchor. Number 3, the template strand is read from the 5' to 3' direction. Yeah, okay, it's read from 3 to 5, but we synthesize in a 5 to 3. We synthesize in 5 to 3 because we only add to the 3' OH end. Okay, good, huh? Last one, telophase unwinds the DNA, and then each one serves as a template. That's okay. Okay, can you write unwind, don't write unzip? Have, because if you go and look back at lessons, right, I may not have said in class, come, let's unzip this DNA. Then now I see in the writing, right, everybody say, I want to unzip the DNA. Yeah? Okay, it's not exactly unzipping. Think about it, the DNA is a helix chain, right? You don't unzip a helix. Okay? You can unzip the zipper, but yeah, unless the DNA came, like that, just like that, two side by side, then you, okay lah, you want to unzip, I have nothing to say. But this one is unwind. Okay, you can unwind. Sorry? Separate, okay, that's also okay. Separate the DNA strands, that's still okay. Okay, next question. Yeah, you're okay with 3, Okay, maybe we just look at this graph, You understand what's happening in this graph. Okay. This is the amount of DNA per cell. Okay. Here we see at this point the amount of DNA increases, it almost doubles. What phase is this? Okay, so S phase is the only phase where the amount of DNA will increase. Right? Can you tell me what's the flat line part? What phase are those phases? The flat line. DNA packaging. Okay, so here is S phase, right? Okay, then what's happening here? Here. Okay, this is S phase. I may say this is G1 maybe. Okay. Then what is this? What is here? Okay, maybe that's G2. Anything else? Prophase, okay. Prophase is possible. So P for prophase. Anything else? Okay, so yeah, and the drop over here is cytokinesis. That means over here I can still have my metaphase, there's still anaphase, there's still telophase. Why is telophase still counted? Because it is still within a single cell. Everything is still within a single cell. So only at cytokinesis at this point over here where they pinch off into two separate cells. Okay, so you must understand what this graph is trying to show you. You could add it to the graph, these additional points. In the notes that you are making or about to make or have already made, actually I will encourage you to draw this graph in. This graph is a very useful graph to show the entire cell cycle, the amount of DNA. I like this graph. That means to say...
No, I'm telling Ida it's recorded so she doesn't know.
Okay. Is this still recording? Okay. Yeah? So this graph is good, I really like this graph. I really like this graph means... Okay, next. This question 4. Question 4, I'm going to start question 4. Understand? Okay, start question 4. Okay, question 4 is an important question. Understand? Yeah, question 4. Let's try to interpret what number of chromosomes and number of DNA molecule means. It means separate things, huh? Okay, I'll show you the first one. This is what it looks like metaphase. Metaphase, and then I have a cell over here. This cell contains 6 chromosomes. So 1, 2, 3, 4, 5, and 6. There are 6 metaphase chromosomes. How many DNA molecules are there? Okay, each sister chromatid is a DNA molecule. So how many DNA molecules are there in total? And see the 12? When I write the 12 out, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So there are 12 DNA molecules. When we go into Anaphase, each one will separate. 1, 2, 3, 4, 5, 6. How many DNA molecules are there now? Okay, correct. It's still 12 DNA molecules, right? 12 DNA molecules. Same number of DNA molecules. No diff, right? Okay. How many chromosomes are there now? Actually 12. Each one of these chromosomes are now called daughter chromosomes. Okay, they are called daughter chromosomes. So there are now 12 daughter chromosomes. Okay? There are 2 types of chromosomes. There's metaphase chromosome and then there's daughter chromosome. They are still both chromosomes because they are highly condensed. The only difference is that one has 2 attached, one is separate. So the amount of DNA remains unchanged, but the number of chromosomes can change. For this particular question, option D in your worksheet says 12 for telophase, right? Okay, so if you had chosen 12 for telophase, actually you're right. Okay, if you chose D, you're right, because the option is wrong. Wait, does that make sense?
What is 6 for telophase though?
Okay, in your worksheet put 12, right? Yeah, that's not correct. It shouldn't be 12 at telophase. This is what it looks like at telophase. So if I go to telophase, you'll find the same cell. You will find 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Nucleus reforming. But is it still within one cell? It's still within one cell. So there are still 12 DNA molecules, but there should be 12. Hey, wait. At the end of telophase and cytokinesis, I forgot there's cytokinesis. So let's split this into half. Okay, at the end of cytokinesis, each cell should have 6 chromosomes and 6 DNA molecules. Yeah? So actually, if you put D as your answer, you are right. If you put D as your answer, you are correct. If you put A as the answer, you are also correct. Because A and D both are not showing correct things. Next, question 5. Okay, question 5 is an important skill for this chapter. We like to see if you can identify the phase of mitosis based on actual diagrams. And this is really quite a skill to pick up. I find it quite fun to try to identify. Wow, this one looks like a lot of ink here. Okay, so let's identify. I'm going to give you the key characteristics of how to identify. Expect to see diagrams like this. Will not provide you the actual drawing anymore. Yeah? So how do you identify? There are some things you can see under the microscope and some you can't. Truth be told, the only thing you can see are chromosomes. Okay? So what can you see under the microscope? How to draw a microscope? Something like that. Okay, so what can you see under a microscope? you can only see chromosome. Okay, this is something you can see. What can't you see? Actually, you cannot see microtubules. Okay, you cannot see microtubules under the microscope. Therefore, this presents us with an interesting problem. If you were to look at a cell that's going through mitosis, all you will see is what you will see in the next question. In the next question, you see all these cells that they drew. Notice there's no microtubule, no centrosome, right? No mitotic spindle. This is what you will see in real life. Mitotic spindle, microtubules are a bit too small to be able to be seen. All you will see are the chromosomes in the real cell. This is a real image here that you're looking at. Okay, how do we identify each phase of the mitosis? Number one, prophase. How to identify prophase? Firstly, you will find worms. Okay, you'll see worms. Distinct worms. Okay, worms, you don't go and tell me that this prophase got see worms. I need to say very clearly. Sometimes students just write what I say. Okay, you'll see worms. Not only that, they tend to be organized in a circular boundary. Notice that the worms seem like there's some imaginary boundary that's keeping them there. Okay, that's because the nuclear envelope is just fragmenting. So that's why they are still in this formation. Imagine. Okay, it's still like that because nuclear envelope just fragmenting. Okay, that's prophase. What does metaphase look like? For metaphase, you tend to find that the chromosomes cluster to the middle. What would that look like? It may not be so obvious. So we look at this drawing over here. Which one might represent metaphase? Okay, usually metaphase, you also find the poles look emptier. Okay, because everyone's coming to the middle, so the poles look rather empty. Okay, out of 1 to 5, which one looks like it might be metaphase? 5? Okay, so actually in real life, this is what metaphase would look like. Okay, let me go to the structure question. Structure question you also see in this diagram here. You look at D. And you see that for D, the chromosomes seem to be in the middle. You see the poles seem rather empty. Yeah? So that's what metaphase looks like. Some of you may be thinking, yeah, I thought you say you cannot see microtubules. How come here it looks like you can see microtubules? Right? Okay, the only reason you can see microtubules here is because these cells have been treated with a special protein that makes the microtubules glow. But without any treatment, actually you cannot see microtubules. Okay? Okay, so we go back. So we've identified metaphase. Next, phase is anaphase. For anaphase, what you will see is the chromosomes pointing to poles. You won't see the typical, very nice order like that going to the opposite poles. Instead, what you will see are the chromosomes just look like single lines. Why do they look like single lines? Because actually what we tend to draw like that tends to be just very, very close to each other. You cannot see it very clearly, the system chromatids. Okay, so out of 1 to 5, which one looks most like anaphase? Yeah? Can you actually see the sister chromatids bent in this fashion? It's not very obvious. Maybe a little bit here and there, but that's the best representation that you can see in real life. This is not microtubules. You cannot see microtubules. So what you're seeing there are chromosomes. Finally, telophase. For telophase, you will find sometimes nuclei reforming. And you should have 2 times of it. Right? You should have 2 nuclei reforming. When the chromosomes decondense, you should also see the chromosomes decondense. What does decondensed chromosome look like? Or rather, what does chromatin look like? So these are chromosomes, this will be chromatin. What does chromatin look like? Well, chromosomes look like worms. Chromatin, actually you cannot see the strings. It will just look like a very dense mass. When you think about it, they are in a human cell, there are 46 chromosomes. If I employ all of them, it just looks like a dense mass. You will not see strings. Okay? So out of 1 to 5, which one might represent telophase? Yeah? 1 to 5, which one looks most like telophase? Yeah, 2? You can still see like some worms, right? But the worms seem to be converging towards a dense mass. That's because all the worms, all the chromosomes are uncoiling, decondensing. If we look at this diagram over here, the real sample, out of A, B, C, and D, which one looks like telophase? C?
C is cytokinesis. It's already the.
Okay, so good observation. Notice for C, it's already formed a furrow. So they already split into 2 cells, cytokinesis.
Alright, there's no
Telophase, sorry.
Uh,
A is still attached.
So what do you think it is? Okay, so this one I would say is like end anaphase, start of telophase. Notice that actually it's very hard to separate the phases very clearly. Everything happens in a flow, one after another immediately. Okay, very finally, there's the last phase, which sometimes you don't talk about, is interphase. What does interphase look like? In interphase, you will find nucleus, because it has not disintegrated. You will also see nucleoli. Okay, and also inside the nucleus, you will just see chromatin. And what will you see as chromatin? You will just see a very, very dense mass. You will not see any distinct lines, just a dense mass. Okay? So what would that look like? Let me see, I think I've Googled it before. If you want to practice, okay, ignore my number of tabs. Okay. If you want to practice, you can go and click cell cycle onion cells, then you have a look. Okay? Right? Look like right now it's anaphase, this metaphase. And then those two are prophase because you can start to see distinct worms forming. We'll consider this interphase because you cannot see any distinct worms. You can still see the nucleus, nucleoli still present. Okay, so if you want to practice, you can go online and find one of these images and you can try to identify it. For example, this is anaphase. You see? Look like anaphase. I would say this one also anaphase. Yeah? Can you find any that's in metaphase? Can you see any cell in metaphase? Bottom? Center? Okay, yeah, this is like metaphase. Yeah? Okay, so it's quite a skill to be able to identify all of them. Let me now show you a video of what mitosis really looks like. Okay, with all the phases one after another melts into each other. Okay, then try to identify the phase. What phase is this? Yeah, it's prophase. You can see the fat worms. poles start to have more empty space. They're trying to go into the middle. Okay, meta, and then do you see any microtubules? You don't, right? They are invisible threads that you cannot really see. Membrane vesicles forming. Okay, that means this is a plant cell. Okay, for plant cells, there will be a row of vesicles that will come to the middle. Okay, then you have two nuclei. So that's what really mitosis looks like. Right, quite a lot of Jun is clapping in excitement, appreciation. Okay, so that's what mitosis looks like. Let's go back to the questions. So if I were to look at question 6, what's what? Okay, sorry, so what's 5? Question 5, this is prophase. Can you see another cell in prophase? Of course I can. Can you see another cell in prophase? Okay. Okay, then what about the rest of the cells? What phase are they in? Okay, there's no distinct worms yet. So it's a phase before prophase, interphase. So actually, if you think about it, right, is it true or not? Remember when we draw the cell cycle, cell spends most of its time in interphase. So that means if you were to look at a large sample, majority of them will be interphase. Only some of them are dividing. We cannot tell. You say G1, you say S phase, say G2, I also cannot for you. Yeah? Interphase is more general. Interphase covers all three. Yeah? Okay, this cell, can you identify each of the phase? Number 1, what's number 1? Ana. What's number 2? Then what's number 3? Okay, that's interphase. Maybe towards the end of interphase going to prophase, so as you can start to see some worms forming. What is 4? Prophase. You can see worms, you don't see the nuclear envelope anymore. And 5 is metaphase. So if you go and sequence that, you should get D as the answer. Okay, personally I enjoy this chapter because I'm a very visual learner. So I like to see all the diagrams, like to identify. This one over here, what would the sequence be? Okay, I would say A is like N, Anaphase, start of Telophase. I would say B is like Prophase. Okay, the chromosomes are starting to be very clear. I would say D is metaphase and C is cytokinesis. So I will line them in this way. B, D, A, C. Good deck. The next question asks you to state the stage D. Stage D is metaphase. Describe the events that occur. The kinetochore microtubules attach to the kinetochore, and then it aligns the chromosomes singly in the middle. Here, I would like to point out a misconception that I noticed when I was marking all your scripts. Okay, there are two interpretations at the moment when I mark your quizzes. The first is this. Okay, we know that the chromosomes need to align in the middle, right? How did they get there? Okay, so you are talking about kinetochores, microtubules. Okay. So I have two options for you. First one, the chromosomes move to the middle. Okay, then the microtubules attach to the kinetochores. Okay, so option 1. Number 1, chromosomes move to middle. Then second step, microtubules attach to kinetochore. It is option A. Option B, microtubules attached to kinetochore. Then the microtubules align the chromosomes in the middle at the metaphase state. Okay, so this one metaphase state. Okay, which one? A or B? Okay, who says A? Chromosomes move to the middle first, then the microtubules swing in to attach to the kinetochore. Or B, the microtubules attach the chromosomes wherever they are, then they slowly pull them to the middle. Which one? B? B is the real one. Okay, so B is what actually happens. The two fishermen from both sides throw out their lines to catch one chromosomes, then they tug of war until the tension is the same. And if the tension is the same but on both sides, then they'll be in the middle. If they find that one person is pulling harder than the other, then it won't be in the middle. So actually that's how they control. They control by tension. Okay, when they feel the tension is just right, everyone will be in the middle. Okay, so B is actually how things happen, not A. You need to attach first, then you can align. Okay, the fish don't jump, the fish don't go into a net and you go, okay. Next, state the stage of mitosis where these cells have arrested. What stage is this? Let's use it as practice. What phase do you think this is? How you practice that? Metaphase? Okay, looks quite lined up. There's a lot of empty space at the poles. You're right, it's metaphase. Suggest how biflastin acts to prevent mitosis. What did biflastin do to result in the cell just stopping at metaphase? So anything that's reasonable, what do you think this drug did to stop the mitosis?
Got rid of the centromere.
Oh, got rid of the centromere. Okay. Centromere actually cannot.
Not centromere, sorry, centriole.
Okay, perhaps the centrioles are affected. Why would you say that?
Because if there's no centriole, then there's no food for the
Okay.
Yeah.
No centriole, no microtubule. No one to really do the tug of war anymore. Fair enough. Maybe the centrioles are affected. Anything else that might be affected? So anything within reason, Anything within reason. I could affect the centrioles. Anything I could affect. Yeah? Maybe paralyze the microtubule. That's interesting.
Go to metaphase, right? So what I said.
Okay, so what's happening in this question is this. He actually managed to go into prophase, then went to metaphase, then they administer the drug. Yeah? And now it's not going into the anaphase anymore. Okay? So, okay, maybe paralyzed microtubule. Anything else? That's within reason. Okay? So these are some examples. Perhaps it can inhibit the formation of the kinetochore microtubules. Wow. Maybe it could prevent the binding of kinetochore microtubules to the centromere. Anything that's reasonable. Yes.
That works because the microtubules already binded in metaphase, right? So that works.
Okay, so maybe prevent binding, maybe it cause it to detach. Detach. Yeah. Is this the last question? Yeah. No, the subsequent ones are all meiosis. Yeah. Affects the microtubule structure. Okay, I see where it inhibits the microtubule structure such that it cannot form the mitosis. But you commit, correct? Alright, okay, so that's your RE for cell cycle. I'll now go into the first few questions for your MCQ. Okay, or whatever time we have. Right. All of you are very tech savvy. You don't know of any method through which I can go through questions with you online. Now, without being lying, because you all have so much to study. What is the story? Sorry? Yeah. Would you all mind if I set a pattern page?
We can do a gem board. Then anyone should answer.
Gem board. I'm trying to think of what's most efficient. So I thought Caitlin, right? You take photo of the question, you put there, then you say I got question here. Then I reply there. Yeah. Because I don't think I will be able to go through everything. Okay, question 1 to 10. Okay, question 1 to 10. Question 3, by Raksha. Question 2, by Bansi. Okay. Question 6. Any more? 6 and 7. Any more? Question 1 to 10 only, yeah? Abi, question 8. Any more? Okay, if you think you got a question, maybe you want to check your friends also, if it's careless or not. Is that all? Question 2, 3, 6, 7, 8? Yeah, we move to question 2. Hey, question 2 proper careless, yeah? Question 5. Caitlin, question 5. Okay, read question 2. Hey, Mansi, know already. Okay, question 2. During the production of apple juice, enzymes are used to break down the components of the cell wall. Which carbohydrate will be produced by this hydrolysis? This hydrolysis means what happens if I break down the component of the cell wall? What is the cell wall made up of? Cellulose. What happens if I break cellulose down? I'll get glucose. So that's why C is the answer. Okay. In question 3, the diagram shows a triglyceride molecule. What happens if I hydrolyze this? Okay, where can I hydrolyze this? I hydrolyze it specifically at the ester linkage, which is here. So I chop this up. Pop. Then I will get one glycerol molecule, and I will get one fatty acid on this end. Let's look at the fatty acid. Because of this double bond, I will consider this an unsaturated fatty acid. So the answer with glycerol plus unsaturated fatty acid will be the answer. Raksha? Okay. Next, question 5. Oh, okay. The diagram shows a part of a DNA molecule. Oh, like this. Okay. So there, these are all your GH3 topics. Question 5, okay? Question 6 and 7. Which is the best explanation for what happens at X? Nikita, what do you choose? D, is it? The number of enzyme substrate complex has reached a maximum. Okay, the activation energy of the reaction has reached a maximum. Actually, the role of enzyme is to lower activation energy. If we increase activation energy, it makes it harder for the reaction to proceed. Yeah? So statement A, actually if you want to say anything, at X, the activation energy actually should be the lowest. Okay? Some of you may have chosen the kinetic energy of the enzyme has reached a maximum. Actually, that's not true. At this point over here is when the enzymes have the highest kinetic energy, when the temperature is the highest. But by this point it's denatured. Okay, so actually kinetic energy is matched with the amount, the temperature. The highest temperature is when the kinetic energy is the most, but it's useless by then. Which is the best explanation for the low rate of reaction? Nikita, what do you choose? D? Okay. I have to say that C and D actually are quite similar in phrasing. So for this one, I think I'll give it a pass. If you chose D or C, I think it's okay. Because both of them are quite similar. Personally, I chose C because this one feels as though they are not, okay. D feels as though they are talking about the entire reaction. They seem to say that the temperature, okay, that the entire reaction will not proceed. But actually, there are many enzymes present. This one, personally, when I read this, I felt like, maybe some enzyme substrate complex can still form. Some don't form. But I admit, after I argue with a lot of students, then it seems quite ambiguous. So both are still quite logical. Okay, so C and D. Question 8. Okay, this is a question on enzymes. Okay, this one you need to recap. Abi, you know already? You know already? Can already? Sure. Okay, come, this question 8. Okay, this is the typical question on inhibitors. So often you'll see a graph of rate of reaction followed by substrate concentration. So let's look at a normal setup. Okay, if I have enzymes and I increase the amount of substrate present, will the rate of reaction increase or decrease? Okay, will the rate of reaction increase if I increase the amount of substrates? Increase, right? It will increase. But it will increase at a decreasing rate. Eventually it will plateau out. Eventually it will reach a point where no matter how much substrate I give, the rate of reaction doesn't increase anymore. Imagine you can only do 3 papers a day. I give you 10 papers, right? It's not going to make a diff anymore. Give you 20 papers, it's not going to make a diff because it's still only able to complete 3 papers a day. Yeah? So that's the idea here. Imagine now I have a competitive inhibitor. This is the active site. This is a substrate. This is the competitive inhibitor. The competitive inhibitor can compete with the substrate for the active site. So you will find that at the very beginning, the rate of reaction will be lower. Because there's this competitive inhibitor out-competing the substrate for the active site. But what happens if I increase the amount of substrate present? If there are more substrate present, I increase the chances of the substrate reacting with the enzyme. And so eventually I can reach the maximum rate again. What about non-competitive inhibition? In non-competitive inhibition, you'll find that there's another site where the non-competitive inhibitor can bind to. But when it binds to it, you'll find the active site totally changes. If it totally changes, now I have some enzymes that are not even working. Imagine now, originally I had 3 working enzymes. 1, 2, 3. If I knock out one, now I have 2 that are functional. So that means to say, no matter how I try, even if I supply more substrates, it doesn't matter. I have one person who's not doing his part. And that will result in this graph. You'll find a graph where the maximum rate of reaction will always be 1 enzyme less. Okay, I should see a crash course on an inhibitor. I'm not sure if that makes sense. Okay? Okay, so it always be 1 enzyme less. Okay, so this is what the graph will look like. That's why for this question, competitive, it should be C. C is the answer. Yes. Yeah.
Does the point of maximum for non-competitive be the same as the substrate concentration?
Say again, what's the what?
Substrate concentration and concentration. Does the point where it reaches maximum be the same?
Oh, so you're saying must it be at this point over here, is it? Not necessarily. Okay, yeah. Okay, that's question 8. Okay, we completed question 1 to 10, please.
I have a question about the test.
Yes, okay, question 10.
Why can't it be A?
Okay, why can't it be A? Amylase is denatured at low pH. That is true. Hey, wait, what cannot be concluded?
Oh, what cannot be concluded?
Yeah, what cannot be concluded?
It cannot be concluded. How do you conclude that it's super denatured?
Yeah, look at the number 2 experiment. When we added hydrochloric acid, the amylase ceased to work.
How do you not denatured? What? It could just be that it doesn't work. The reaction doesn't work.
Oh, okay. So you're saying that perhaps it is due to now the burette test not working. Sorry, the Benedict's test.
Also, for example, this is optimal pH of amylase is 7, then it's like you cannot conclude. But then you cannot conclude that it's denatured. It could just be that low pH is not optimal, right?
Yeah, it's not optimal because
But how do you know it's denatured?
that's the only thing that can happen.
Why? It can just like
It's not even if it's
Oh, okay.
not optimal.
You know what? Low pH means it's acidic.
yeah.
Yeah. So,
If it's not optimal, right, then there will be it doesn't make you mean there's really no reaction happening.
so are you saying, okay, are you saying that it's just not optimal?
Yeah.
But it may not be
Like
denatured.
for example, when there's a difference in temperature, sometimes it's not optimal, but then it's not denatured, right?
Oh, okay. Understand. Same idea?
Yeah, because the graph, right, it's
Yeah.
not zero yet for the unless amylase is a straight line.
Yeah.
Yeah.
So your issue with this statement is that if they wrote that this is not the optimal pH, you'll be happier with it.
Oh, maybe that's the answer because even if it's not optimal, then it would have been like green or like lower concentration. But the fact that there's nothing produced says that it's
Hmm.
denatured.
See, your explanation seems quite fine.
Hey, listen to me.
Which is the question also what? Say.
Because if you say this is not optimal, right? This is not optimal, something is wrong.
Okay, anyway, the graph of pH, right, you'll find it looks like that. Right? So that means even past the optimal pH, there will be still some reaction taking place. Yeah?
Like over there it would have been green, and then slowly, then zero is green.
Yeah. Actually, I like. Okay, so perhaps what would be better is that maybe amylase, some of the amylase is denatured at low pH. Because I'll have to admit it won't be all of the amylase that's denatured. That's true also lah. So that means with the fact that we see that it's totally blue, that means all of them is denatured. Nico? Why is B correct? The digestion of starch is due to amylase. How do I know it's digestion, is it? Okay, this is a Benedict's test. So Benedict's test tests for presence of reducing sugar. If what is starch made up of? It's made up of glucose. So if starch is digested, you will get a positive Benedict's test. Notice that the first one, amylase plus starch plus water, you get orange precipitate. That means some digestion took place. Second one, plus the acid, now we know that the enzyme played a role.
I get it.
Yeah. Okay, last one, just water and starch. No. Yeah, that means if you don't break down starch, you won't get to see the observation. Okay? Alright, not much time left. 11. I'm going to assume some of you need 11. Okay? 11. Anyone chose A? Okay, A, you read carefully. Lactase is not present to keep the peptide bond between the monomers. Lactose is a sugar. Okay, lactose is a sugar. Therefore, it's held together by glycosidic bond, not peptide bond. Okay, then now the question B and C. Both B and C seem quite similar. Both seem logical. I would say actually both are logical. But the reason why I chose B as the answer is because what's the function of enzyme? lower activation energy. C implies that there's insufficient energy for the reaction to proceed. Almost implies that the solution to it will be to give it energy. But that's not a function of enzymes. Enzymes don't give energy, it lowers the activation energy. So that's why I chose B at the end of the day. C still makes some sense, but doesn't quite bring out the essence of the function of the enzyme. Okay? So question 11. We'll stop here. Okay, still got time. Question 12. Anyone needs 12?
Question for the what's the difference between A and
A and D. Oh, that's okay. D, I don't know what D exactly is, but it could be a lysosome. A, I would say that whatever A is, it just came out of the Golgi apparatus. So it's some form of secretory vesicle. Okay? So which of the pathway shows to a transmembrane protein? The answer is, Basi, what do you choose? C, GFB. So G, F, and B. Okay, what is G? Free ribosomes. A transmembrane protein is synthesized by bound ribosomes, not free ribosomes. Okay? Not proteins used by the cell. Proteins that do not require to be held within a vesicle or embedded in a membrane will be synthesized by free ribosomes. Anything that requires to be embedded in a membrane or be kept within vesicles needs to go through the RER, because then it can be packaged in a vesicle. So an example of a protein that will be synthesized by G, free ribosomes, will be maybe hemoglobin, because that's used within the cell. Lysosomal enzymes will need to go through the RER, because I need to package it in a vesicle. Okay? Membrane proteins also need to be packaged in a vesicle, or rather on the membrane of the vesicle.
Why is nucleus not included?
Why is nucleus not included in the pathway? Because the nucleus isn't the one that synthesizes the protein.
But it produces ribosome.
Yeah, it does. It does. So which answer are you looking at? But in reality, it is part of the pathway, but it's really, really early on in the pathway. Not even considered really part of protein synthesis yet. Okay? Okay, I think that's all. We'll start at question 12. Next round, I see you. Please try to get all the MCQs done. We will go rather fast. Whatever I cannot go through, we'll go through via Padlet. Or maybe let me set up the Padlet ASAP. You can already start to post it there. And then I'll try to answer along the way. If you have no time to complete your structured questions, may I advise you to just try to write in bullet points. You must try. Wait, come in. Zoe, could you help me stop the video?